but no one else would.
Right, yeah.
Most people say my sympathy.
Right.
So welcome to this next, this year's seminar. So I'm very pleased that Professor DiCarlo from MIT agreed to join us today to give us this presentation. Seeing this part of this work at ICCP last year, that's our major computer vision conference. And this is certainly not just computer vision. It's in fact work on the base of computer vision and neuroscience. And I was really impressed by what these people, in particular, this group has been able to achieve in recent years. So I hope you're also going to enjoy it. And so without further ado, we'll leave the stage to Professor DiCarlo.
Okay. Thank you for having me. Thank you. Everybody hear me okay back there? Okay. So again, thank you for having me. My lab is really sort of a systems neuroscience lab, but we're interested in problems that connect to engineering, and computer vision problems, especially object recognition, is really one of our kind of lofty goals if we can inform that community. So I really appreciate the chance to try to explain how we think our work might connect to the work, especially on the vision side, going on in this group. And I know this group is bigger than just vision. It's also language and speech processing. And I don't know if I'll say anything direct about language and speech. In fact, I won't. But hopefully you'll see some ideas on the computer vision side of how neuroscience is at least connecting to that problem. That's my major goal today, is to try to explain our view on that to you. So let me sort of get started by giving you sort of a perspective on the way, as engineers, we like to think about this problem here. And I mean, I should say also, for those of you who know me, or some of you don't know me, I trained as a biomedical engineer here at Hopkins, so I sort of have a special attachment to this place. And part of what I learned from my PhD advisor, the late Ken Johnson, who many of you may have known, worked here right in this building next door, was to always take an engineering goal-oriented approach to problems. And one of the problems that Ken would help inspire me to think about is the problem of what I'm going to refer to as object recognition, and why that problem is interesting. So I'll take you through that in a lot of the talk here. But let me sort of back up and give you a big picture and to say, look, if we want to understand about things that the brain does, if we want to use the brain to inform better engineering or better machines, it helps to step back and look around and go, well, where does the brain actually excel relative to machines? Because there are many domains, and some of them are listed there. I'm sorry, is there a pointer nearby? Does anybody have can I? No, I'll just sort of hand wave. Let me see if I can use my... No, I can't use my mouse. I'll just kind of point up here. So there's many ways that you can actually succeed as a machine. Things like calculation, chess. We saw Watson just won at Jeopardy. Lots of forms of memory in a simple form. Machines have, this machine right here has way more memory in what it can recall in detail than our brains, I would argue. But in terms of complicated things that require generalization, and seeing is one of those, a broad category here, there are things like pattern matching, which doesn't require much generalization, which machines have also won. At barcode readers at your grocery store, an example of that. But object recognition, we still are, we think, succeed in that over machines. And really the goal of my lab is to discover how the brain solves this problem in the form, not just to speak to neuroscientists, but again to computer vision. And our goal really practically is to help move that column over there to say at some point we'll have machines that will match us and will almost certainly supersede us at some point. So this is our sort of operational goal right here. And from a neuroscience perspective, we view this as a gateway problem. That is not just an engineering problem of object recognition, but a problem of how vision works more broadly and how neocortex works in a way that might generalize to other sensory systems, for instance, somatosensation or audition. So let me give you, this is sort of an overview of what I want to try to say today. I'm going to talk about the nature of the object recognition problem. I'll spend most of the time talking about what I'll call phenomenology, that is neural codes that we can extract at high levels of the visual system, and how we think they explain or fully account for object recognition performance in humans. And then near the end, I'll take you through the Holy Grail question and how we're thinking about that, which is what are the underlying mechanisms that transform the image to a representation, to set some neural codes, the phenomenology here, what are those mechanisms? And that's really the Holy Grail. We don't know the answer to that question, and I'll tell you a bit about how my lab and others are trying to approach that. But that will just be a brief outline. So the big picture here, object recognition, this means many things to many people. The way I like to set up this problem is to say, well, look, in the real world, it's intuitive that you have the scene. You don't take the scene all in at once. And in fact, the part of the brain that we work on, the ventral stream, sort of really focuses on the central, so let's say 10 degrees of vision. So that's about 2 hands at arm's length, those of you in the room. So that's sort of the region that's most sort of dominantly processed by the ventral visual stream that I'll talk about in a moment in the primate. And so that's sort of approximated here. So you might sort of say, well, there's one or more objects in that region. In this case, you might want to utter the word car if you're a recognition system. Humans don't, of course, just stare at the center of this, but they make many rapid ballistic eye movements around called saccades, and they dwell at each point for about 200 milliseconds. And what that brings to their ventral stream engine is something that looks kind of like this. Sort of a series of snapshots. You can think about the ventral stream receiving those inputs as the eyes are driven around the scene there. And again, you could recognize one or more objects in each and every one of those inputs, even without notion of where you were going to go in that scene. I think you could say, oh, there was a sign, there was a person, there was a car. And that intuition, that's what we call core object recognition, what I call the central 10 degrees of visual field, about 200 millisecond viewing duration, which is the sort of typical saccade minimum fixation interval. That's the engineering or sort of top-down constraint that we want to apply to this problem. So trying to bring all the way from scene recognition to something that's actually operationalizable that we can study in the lab. So this is what we call core object recognition. Intuitively, you feel we're very good at this. This is a nice demo of this I like to show at all my talks. It's basically a movie. This is 6 frames per second. You can notice that you can recognize one or more objects in each and every one of those frames. And you notice that there's no sort of pre-queuing. It sort of feels effortless. You do this, again, very quickly, less than 200 milliseconds. I don't need to tell you kind of what you're going to see. You can entertain many objects sort of simultaneously at once, it feels like. And most importantly, your ability to do this is highly tolerant to variation. For instance, the illumination conditions on Yoda here. And that turns out to be the crux computational problem, as many of you know, and so I'll spend a great deal of time talking about that. Okay, so here's the system, the human brain, that you guys just used in your head to seemingly effortlessly solve that problem, a problem that still is a challenge for machines. And so we'd like to understand how this system does it. That system is hard to study directly, but we use this model system. This is the primate visual system. This is not shown to scale here. And the reason we like this primate brain system is that we know a lot about the primate brain. For instance, we know this ventral visual stream here, shown in yellow, is deeply involved in object recognition. And this is known from the work of many labs, including Ed Connor's lab. Ed is here in the audience. And so we know a lot about the primate brain in this part, and so we know kind of where to look for the solutions. And we can go in and study those representations at the most appropriate level of abstraction. Just to orient you, the ventral visual stream has a series of visual areas, V1, V2, V4, IT, shown here. I'll show those again later. And IT, which is sort of the top of the ventral stream, the area I'll talk about today, it projects to regions involved in what are thought to be decision and action and storage or long-term memory through projections as I've schematized here for you, just to orient you on the brain. And again, the monkey brain, because this is a monkey and not a human, we can go in with things like extracellular electrodes or electrode arrays and go and record neural activity, again, at the level of spiking activity that we think is the most appropriate level at which information is passed around the ventral stream, and that's the level at which we want to extract signals. So we can do that in a monkey. We know a lot about the visual system of the monkey. Okay, so here is as an engineering diagram, here is the ventral stream of these tangled cortical sheets that had to be stuffed into a skull. Here are them unlaid, sort of unwrapped as sort of schematic sheets of tissue. So there's a series of visual areas here, the retinal ganglion cell layer in the eye, the LGN in the middle of the brain in the thalamus, area V1, the first cortical visual processing area in the back of the brain, V2, V4, and again this higher area IT. And here they're schematized as sort of millions of neurons in each area. They're schematized as a sort of feed-forward flow of information, intracortical processing, and also feedback flow of information through those schematized arrows. And so that's the big picture of how we see the big picture layout of the system. And what our lab does is consider each stage along the ventral stream as a new population representation. Okay, so the reason we argue for this, and that others have argued for this, is that there's a new sort of retinotopic field. There's a map of visual space at each of these visual areas. So there's a retinotopic map in the retinal ganglion cells. Again, in LGN, V1, V2, and V4. And IT, things get a bit more fuzzy and unclear whether IT is a single area or not. But let's just sort of leave it with question marks as now, and I'll just talk about it as IT for today's talk. Okay, so that's the sort of big picture layout to bring you up to the primate brain. So what happens in the primate brain is if you see this image, what you saw in that movie, the image was shown to your eyes, it produces essentially a photographic copy on the back of your retina. Ken Johnson would call this an isomorphic representation. So it's a one-to-one mapping almost of what's out there in the world. It's just been transduced. Okay, there's some nice processing and gain control and things that you would put in a nice camera, but it's from a point of view of pixel arrangements, it's largely isomorphic. Now it gets processed very rapidly through this series of visual areas in a way that the latency suggests, 60 milliseconds, 70, 80, 90, 100 milliseconds. So in about 100 milliseconds, you get patterns of firing of neural activity here schematized by little red dots, active neurons in IT cortex. That pattern of activity looks nothing like this image. It's not just a copy of the image. It's a transformed version. So a new image, new transformed representation in IT, new population pattern. And when you see this movie that you watched here, what your IT is clicking along at a lag of about 100, 150 milliseconds following this, just producing a new representation each time an image comes up. And it can follow easily at rates like this here, 6 frames per second. Okay, so this is the sort of layout of the system, and the algorithms that produce this transformation, which again from here to here is all cortical algorithms. Those algorithms are really unknown, but we think again they have remarkable potential if we can understand them in the context of this problem. Maybe that might generalize to how we think about cortex more broadly. Okay, so that's the big picture view. My lab has a number of techniques that we use to try to kind of attack this ventral stream and this problem of recognition. I won't talk about all of them today. I'll just sort of list them here: behavior, physiology, computational methods, some functional imaging, and interventional techniques that I'll only allude to. So we have this sort of this set of tools, but our major questions are really how does the brain transform the image to this new magic population representation in IT? And really that's, again, what we consider to be the Holy Grail. It's how does the brain transform this? And we like to think about that in the context of a canonical cortical computation. But for purposes of today's talk, this problem, by the way, is not yet solved, and that's what, again, our Holy Grail. For the purposes of today's talk, what I'm going to try to do is talk about, step back and say, what do we know about the nature of the solution, which I'll call the transform result at the level of IT cortex, at the top of the ventral stream? Where is that located? And again, I've already sort of told you there's something in IT, but is that IT solution sufficient to explain the kind of problems that we're interested in this case, object recognition? And now, even to talk about those, we need to step back in sort of a David Marr sense and say, why does the brain actually need to transform the image in the first place? Why doesn't it work right here? What is this whole thing for? Right? And that's really a kind of an engineering or computer vision perspective. So I'm going to start a bit of that. And of course, when you're going to transform things, the reason the brain does this is to somehow guide useful behavior to survive in the world. But we don't know exactly yet how that happens. And most of my talk will be geared at sort of showing you how these representations are well suited to support object recognition behavior. So the behavior that we're interested in here is, again, object recognition. And what makes this a challenge is that if you want to recognize a car or any object, that object can produce an essentially infinite number of images on your retina due to variation parameters. So things like position, size, pose, illumination. Some of these are nuisance parameters. Some of them are interesting other latent variables that you want to keep track of. But from the point of view of saying it's a car, all of these are images of cars. There are many different types of cars, so there's interclass variation. And of course, there's sort of background clutter and potentially occlusion as well. So all of this variation is well known to be the problem that's challenging from a computer vision perspective to say, hey, there's a car in this image, or even a part of a car, or whatever level you want to abstract this to. So this ability is to tolerate this. We call this invariant object recognition, and I say invariant in quotes because that mathematically implies perfect, and we're not perfect at this, as you'll see. So you'll sometimes hear me say tolerant object recognition. So we have some ability to tolerate this kind of variation. And so let me give you our perspective, and this is our geometric, this is the way we like to think about this problem because I think it gives intuition about why you need a transformation. It helps neuroscientists at least think about the kinds of things that have to happen and reminds us that the focus is not so much on sort of creating information but sort of restoring or reformatting the existing information that comes in at your retina. And at least that's our perspective. So you have this, the way I like to think about this is in a state space where each of these axes is the activity of a neuron. Say we'll think about the population representation at the retinal ganglion cell layer. So each of these axes is the activity of a retinal ganglion cell. There are roughly 1 million dimensions here, so a 1 million dimensional space. And so what I want to kind of convey to you is the nature of this representation and why it's challenging for object recognition. So the way you think about this is that you have an image of, let's say, this person who has identity named Joe, who is under one particular set of viewing parameters, pose, illumination, position, scale, and so forth. And that is one photograph of Joe, and so that's one point in this 1 million dimensional space. If Joe undergoes a transformation, such as, for instance, a change in his pose, so that's 1 degree of freedom, he's going to change the image that's being presented to your retina. These activity of these neurons are going to shift, but it's going to sweep out a continuous one-dimensional line because you're just changing one underlying parameter here. So you'll get these other images, but they'll be nicely connected in this way, and that's sort of other images that also correspond to Joe, other points in the representation. Now, if you can imagine, here's a 2nd degree, here's 2 degrees of freedom now for Joe, and you can imagine this is just 2 degrees of freedom, but you can imagine other latent variables that all of these points that Joe could potentially produce in this space is this thin sheet of points that's highly curved up called Joe's identity manifold. Okay, so that's kind of how Joe is presented into the early part of the visual system. Yes? Yeah.
So if you wanted to, or if I wanted to measure this in the primate brain, would I sort of show it 20 millisecond pictures of this with enough time in between so that things reset, or would I show it a movie? Would the results be different?
So you're asking about temporal effects now, right? So the way I'm thinking about this is just still we're doing static images, and I'm sort of keying off, and I sort of introduced at the beginning of core recognition, right? So you make these ballistic eye movements around a static scene. We can think of those as a series of photographs. That's not all of vision, but that's largely what I'm going to talk about today. And yeah, so here if you wanted to measure this, you would present images of Joe in different poses and so forth, right? Yes. And we're talking about now, again, the time aspect of things, right? That's that when you say you're...
What I'm wondering is that this picture is probably, it doesn't matter whether it's, whether you're looking at several time slices next to each other or really far apart, as long as, let's say, like the lighting conditions are more or less the same, because we have adaptations to the level of light, but maybe at the level of the retina, there's not much more than that.
I think you're again referring to if we were showing movies of Joe rather than still images of Joe. This is not well understood, and we in the lab have sort of stayed away from that for the most part. We think of using time, especially in the context of setting up a representation and learning to build it. But you know, I can show you operation that you can do the task I showed you, and so that's operationally the task that we focus on. That doesn't mean that, you know, it is interesting to ask what are the time mechanisms that humans use to add to their inference that Joe's in the scene. But the fact that I can flash an image up and you can say Joe, that's an interesting behavior to me, and that's where we're trying to understand. So I can't say more to your question. I'm not saying it's not interesting. I'm just trying to keep everyone focused on where we are. Yes.
Joe's identity manifold also include all the backgrounds that Joe might be?
Yeah, so that's the part that, so this is a good audience. So that's the part that I think this picture is sort of not, that leaves out, right? So that would kind of be like a fuzz around this, right? Because sort of you can essentially fill the space around this manifold by changing the background around. It's not going to be a simple low-dimensional 6 underlying parameters, right? Well, yeah, I don't like to invoke that because that sounds a little bit like hand of God reaches in and saves us, right? I mean, I was just trying to give a feel for how the data might lay out. But you're making a great point, which is that, yeah, this is nice to think about with the latent parameters of position, size, and pose and the controllable things. But with regard to background, it's not clear, you know, this is going to fuzz up this manifold, and it's not clear how much impact that's going to have. And we are taking inspiration from like these simpler latent parameters. Say, if we think about those, maybe that background thing will turn out to be less of a problem than it initially appears, right? So I'm lumping it in, but I don't know whether this view will actually generalize across the backgrounds, as you've rightly pointed out. Okay, so just to kind of keep moving, I want to give you the idea that a representation that we would call good, this is how we think about good, is if you had a manifold for Joe and Sam, again, some low-dimensional manifold that's curved, but it doesn't matter if your task is to say behaviorally this is Joe and not Sam, and you have a toolbox of reading this representation, which is a separating hyperplane, which to us, we like separating hyperplanes because they're basically implemented as linear classifiers, things we know how to implement, and they're approximately what a perceptron-like downstream neuron could do. So in any case, there's something that engineers easily know how to do. There's something that we think neurons can implement, and so the way we think about the problem is one of producing a representation where now it's easy to decode this variable, perhaps other variables as well. Okay, so this is just to remind you of why we're using kind of linear classifiers as sort of our readout tool. And what I want to point out is that this is, of course, not true. Some representational layouts will be like this, where you know your linear classifier, either because of limited training data or because of the nature of the representation as shown here, will not be able to find that separating hyperplane, and so you end up with an ineffective separating hyperplane. Effectively, you get low performance on a task. And so this is, the way I frame this is a representational problem. This representation is not as well suited as this representation, which I call explicit object representation, in that it's easy to decode with these kind of tools. And so the way we think about the ventral stream is that this is actually a simulation of Joe and Sam three-dimensional objects. Now this is viewed in 3 dimensions. This was a 14,000 dimensional space that we had to project down, but you should think of these as 2 crumpled sheets of paper that under even basic viewing parameters, even without background clutter, cause these things to be highly sort of intermixed with each other, which we call tangling. It's not that they're actually fused. There's always information to say whether it's Joe or Sam in the image. It's about the format of that information. So it's kind of tangled up in this space. You try to put a classifier through it, and you're in big trouble. You can't easily do that. Whereas out here we think there's a transformation, whereas you get some kind of representation with a limited, fewer number of training examples. It's easy to find a separating hyperplane to say the manifolds may look more like this. So you can easily say, let's say I want to say whether this is Joe or not, or Sam, which would be like a shape or identity variable, but as well as other information that's still preserved. And I mentioned that some other latent variables such as position and scale, it's not that they've been collapsed and thrown away, they've just been sort of nicely arranged for you. I mean, this is really trying to make the latent variables explicit. And that's what we think is happening along the ventral stream. My talk's going to focus exclusively today on shape, although we are looking at these other variables as well, for those of you who are interested in that. Okay, so this is the big picture of how we think about the problem and how we think it relates to the ventral stream. Okay, so we call this an explicit untangled object information, and in a computer vision language you could say this is a poor set of features and that's somehow a nice set of features, and there's some nonlinear features on the image space and we don't know what they are, but if we could give them to you or hand them to you, then we think you're going to have a good representation that you could apply to an image. And that's what we would like to try to hand to engineers, as a description of what are those features. Okay, what is that representation is another way to say the same thing. Okay, so let's talk about that representation itself measured directly. So we go and measure those neural codes or those neuronal features and ask, can it really do the thing that, Jim, you're saying here that it can do in a qualitative conceptual sense? How well can it do that? Does it account for the things that we think are interesting? So that's what I'll spend essentially most of my talk talking about. Okay, yes, good. That's the way I'm thinking about the maximal dimensions, but of course you could have redundancy in the neurons and it could effectively be loaded. So how complicated? Well, you could, maybe folks here could help me formalize what would a measure of complexity be, but the dimensionality that I'm thinking about is, again, limited at the upper end by the number of units in the area, and that was a sort of population. So 1 million at the input, sort of 10 million for output for a place like IT. Okay, so 30 million output roughly for V1. So of those numbers is the sort of upper bounds. And of course, there's structure that we know about from neuroscience. As you say, the neurons have receptive fields. They live next to each other. So there's some, it's not as if these are sort of completely independent dimensions, right? And again, I'm giving a very top-level view of the problem and why you need to transform it. And the kind of things that you're talking about are important to understanding it. And I'm not getting into them here today, but I'm not dismissing them either. Stable with respect to viewing parameters like pose or position. Is that what you mean? Okay, so we're talking about time again, right? So I guess I would say that, for instance, we might, let me back up a bit. So we might,
If I rephrase my question, is that probably in the reticulae, you're expecting more dynamic coding and you have more stable coding in IT?
well, stability implies, you mean something like, it's stable with respect to a classifier readout trying to say identity. So that doesn't mean that different neurons aren't going to become active as a face moves across a scene, right? But those individual neurons are coding in a way that they've orthogonalized the latent variables, so which leads to manifolds that look like that, which means that I can put an easy classifier and say, there's a stable read of the population, but the population itself can still be varying as you change the image, like change the position or pose of Joe, right? So it's just a reformat. It doesn't speak to whether it will be more or less dynamic. We could measure such things if we got a number that you would like us to seek. I don't know the answer to that question offhand. What is the amount? It depends on the conditions of how quickly Joe's moving. I can make a guess based on existing data. But I don't know if I've answered your question or not. Well, okay, that's an let's get through the rest of the talk, and we'll see if I don't get on your notion of stability from perception, because I'm going to talk about measuring performance in a moment from humans. Okay, so let me just, again, big picture, for those of you who don't think about neuroscientists, there's sort of three ways to study a system like this. There's a sort of standard way, which is called encoding, and if you read papers that say, here's what a V1 neuron does, then you're trying to map between its image and its response. And ultimately, that's what I said we'd like to do for the whole ventral stream. But that gets challenging to try to take it deeper through a nonlinear system. So you do this. Another way to study it is think about measuring transformations. So for instance, what happens between V1 and V2 or V4 and IT? And Ed Connor's lab has some nice work on that, for instance. So trying to measure what the change is across different levels to try to take the system apart. And then the third way, which again is inspired by originally by Vernon Mountcastle, and then later again by Ken Johnson, who was my PhD advisor, is to build maps between neural activity, which I'll call neural codes, and the behavior of interest, which in our case is object recognition. And this has been applied in simpler domains. For instance, Ken and Ed did this for roughness and touch. Here we're trying to do a similar strategy. Again, neurons, high level of the system, to behavior. In this case, object recognition behavior that I'll show you in a moment. So I just want to give you a backdrop for the study that I'm going to show you next. Okay, so there's lots of ways to study this. Here's the way we're going to talk about it now. And this approach really begins with way back in 2005. This is Tommy Poggio and a couple of postdocs, Gabriel Kreiman and Xiao Hong and I, showed that consistent with other work that you see cited here, that if you record neurons in IT, they have properties that seem to suggest that they can do interesting object recognition behavior. This had already been suggested in the literature. We formalized this with decoder tools in 2005 just to show that you can get a lot of information about these neurons for things like recognition. And the way that I want to connect you to the notion of kind of what these neurons feel like. So if you record from an IT neuron, this is just to give you a sense of the elemental data. You're showing images to the animal. The animal's awake and fixating, so just passively fixating, and images flashed up, 100 millisecond duration image, similar to the movie that you saw, random sequence of images. Each of these tick marks is a spike, an action potential out of the neuron, a response. Each row is a repeated presentation. This is all randomly interleaved. What you should do with your eye is sort of integrate and say, look, there's a lot of spikes over here to this image, so this neuron seems to like this image, and some here, and less here, and less and less. Right? So there's some selectivity, at least across the images here. Now, for those that are following, this is almost surely true for a retinal ganglion cell. This doesn't yet say anything interesting about the form of the information. It just says there's selectivity across different images. Different sites in IT might have different selectivity for different images, sort of highlighted here. Okay, this is just to give you a feel for what these neurons look like in the raw data form. A wave fixating monkey, and all we did was to count spikes in windows. So there are many codes you could try to read out of these neurons, inner spike interval codes, all kinds of fancy timing codes. We just did spike counts in windows. It's a time window that we can later vary, which we consider to be behaviorally constrained, meaning its maximum time was no longer than the animal's early reaction time. So we know if we believe in a causal system to produce the animal's response, we can't have this window go out for infinity. But so we picked windows that were roughly 100 milliseconds, beginning at the latency of IT neurons, which is about 100 milliseconds. So this is all the data I'm going to talk about today from the brain are analyzed in this way, counting spikes in a window of roughly 100 milliseconds. Again, we vary that, and those are lots of details I could take you through, but that's the way to just keep the top-level view. Here's a simulation of a bunch of neurons, again, to give you the intuition. One image comes in. We collected a bunch of neurons in this study, and we can vary the number of neurons that we include in the code read, as we call it. We count neurons, count spikes from each neuron in a behaviorally constrained window, as I just showed you. That produces a scalar value for each neuron. So you have n neurons. You have a point in an n-dimensional space. So you can think of this now. Here is an actual measure of the space I referred earlier. This is not all of IT. It's a sample of IT. Each axis is a neuron. This is one image, so it's one dot in this space. There's some noise associated with repeated trials that I've sort of illustrated with the cloud there. But then the idea is that if you measure many faces or many different views of this face, or that they're going to lead to different points that we can measure, and then many non-faces here shown as red. And what we're asking is, can we put linear decoders on this to say, is this a face or not across transformations? And that's schematized here. This is a schematic data. We did this for a series in this study. This was 8 categories that we had picked ahead of time. They didn't even have any backgrounds on them. This was a very simple first-pass study. But we showed that it was very interesting that just a couple hundred neurons out of IT, you could easily decode real time. You could say which object there was with near perfect performance. And more interestingly, you could do this in a way that would generalize across at least position and scale, and then later against some clutter. So that's implying some sort of ability to, again, these manifolds have been untangled a bit at the level of IT, at least with respect to some of these variables here. And we showed that this was not true in earlier visual cortex, as I already implied earlier in my talk. So we measured that directly, applied these decoding tools, and this was nice. It was cool phenomenology. But this was just, to me, the beginning, right? If you really want to say these codes are supporting recognition, you have to do something stronger than that. And so what we tried to do, and what we've been doing the last couple of years, and this is what I want to tell you guys about, is we've been trying to ask, are these codes, these sort of decodes from rate responses of IT neurons in these population codes in IT, are they, in the monkey, are they sufficient to fully account for recognition? I just showed you could get good performance, 100% correct, reasonable generalization across position and scale, but are they sufficient to account for recognition behavior? Okay, and to do that requires, and this was, by the way, done by Najim Majaj, a postdoc, Ha Hung, a graduate student, Ethan Solomon, an undergraduate. And to do this, what we had to do is, of course, measure first behavior in many object recognition tasks. We had to quantify the thing we wanted to explain more precisely. Then we take the images that we use, the same images that we test humans on, we test those images, present them to the monkey, measure a large sample of neuronal population spiking responses, build putative neural codes, that is decoders off of places like IT, or also computer vision models showing these same images, and then we use those to ask, are any of these codes, these feature sets, sufficient to explain this behavioral performance? This was the game here, right? We have these images, we think we have a good representation, let's test it harder to see if and when it breaks, and that also, in the language, again, of Vernon Mountcastle, would help us to figure out what is the actual underlying neuronal substrate that supports recognition. So it's trying to discover the code by falsifying many codes and leaving some codes still standing. So hopefully that'll become clear as I take you through the details, as if that was a little bit fast. So the idea is this. We're seeking a quantitative link between neural activity and behavior, as I alluded to earlier. And when we say quantitative link, what you'd like to do is say, well, you'd like to do that in humans. As I referred to earlier, we'd like to sort of go into some human analog of IT, show some images, measure them, and then compare with human judgments of, say, is this a face or not? Now, as I've already alluded to, that's really hard to do in humans because we can't go in and access the data at the level of spiking activity. Okay, so this is not the way we thought we wanted to go. Then you say the next obvious thing is, well, we should just measure monkey neurons like we can do easily, and then get the monkey to do a bunch of behaviors and then try to make the link that way. Now, this has its own set of problems because, we could say, well, the monkey has to be trained forever to do the task. It's overlearned. The representations we're looking at weren't actually there at the beginning. There's this sort of problem of doing the study this way. And so what we did was actually what Vernon and Ken and Ed and others had done in other domains, was to say we're going to measure things in the monkey and we're going to compare it with human judgments under an assumption of some species homology, that there's some common substrate in these two, but we can easily get humans to make judgments on these images. Now, this is a point, I'm bringing this up because it's a point of contention often when I present this, so I'd be happy to hear your discussion. The idea is that, well, humans, this should never work because object recognition is somehow too cognitive or requires feature attention or semantic categories or things that aren't somehow going to exist in the monkey brain, or at least not the way we're measuring it. That's a fair hypothesis, but our approach was to say, let's just try, and if it doesn't work, then we'll say maybe that's the reason. But we were going to try the null statement of how far do we get by just comparing IT codes with human performance and recognition tasks. So that's what we did. And so here's what, and partly because it was doable. And so here is, again, monkey ventral stream neurons. We're going to compare to human behavior. Now, what images are we going to use? I said, well, we're going to do some images. What do we use? So you saw before we use these kind of isolated objects on a blank background. We wanted to be more inspired by things that are hard for computer vision. And we also wanted to be able to measure behavior, and we wanted to be able to measure other latent parameters. And we wanted to sort of have this be a kind of a strong Turing test for object recognition. Now, there's no right answer to this that gets into computer vision problems of what is the right data set to do. But given we had to get going on an experiment, we made some choices, and here's what they are. We keyed off the idea that invariance is the hard problem, and so we basically constructed synthetic data sets where we take known 3D objects, where we can render them with known latent parameters of position, size, and pose, and so we can ray trace them, make some object. This is one particular image of a car. Then we wanted to make it more difficult by placing it on complicated backgrounds. We could do this in a contextually consistent way, but again, we didn't want any kind of ways of using that context. We wanted to focus on the object itself, so we did it in a more arbitrary way, where we have random backgrounds and random draws out of the latent parameter set to create images that might look something like this. And so this was our kind of trying to walk the line between making it challenging but still controllable and understandable. Again, a nice point of discussion that I started this morning with some folks. So, this can lead to some crazy looking images like this. Here's the top of a car floating on the background here. The subject would maybe have to still judge that it's a car. But you can see, giving you a feel for it, we can create lots of images this way. It's parametric, and we call it natural-ish. We know it's challenging for computer vision systems still, and we like that. And we said, let's go with this set. So that was what we call version 1.0. Here's to give you a sense of object recognition task 1.0. I won't even tell you categories yet. Let's just see how well you can do. See what that was? Car. It was a plane, right? Okay. Yeah, so that was a little longer than 200. But you can see this is, I don't have to instruct you. I can get, at the end, I'll show you how we did the task. But you do this very well, right? And it doesn't matter that the backgrounds aren't correlated. You can do this, right? And that's really interesting to us. And that's what I said at the beginning. So that's the kind of task that we ask humans to do. Again, I call this object recognition 1.0 because I want to be humble and say this is not the full Turing test of everything you associate with object recognition. It's just our first attempt to get going on quantifying and operationalizing things. So there should be a 2.0 and a 3.0. So this is 1.0. And so what we want to do first is measure human behavior. And here are our measurements of human behavior. I'll show you details in a moment, but just to give you a top-level view, we had basically designed, given the object sets that we used, what you see here is a basic task where humans were asked to say, which of these 8 were presented. That was kind of the task you were doing. Is this a car? Is it a chair? Is it a face? Is it a fruit? What you see, and you see subordinate tasks like which car is it, which face is it, so that's more of a face identity task, and you have amount of variation where we kind of control how much of the draw, the variation of those latent parameters we allow, and I'll give you a feel for that, ranging from very low to very high. The color indicates D prime, so high D prime, 5 is a very, very good, near 100% almost correct performance. 0 means near chance. Okay, so you can see these are actually the mean human data values that we collected. The way we did this was humans, they got about 100 millisecond viewing time, as you saw. They were asked to make, after the image, an 8-way choice of which object category they saw. The images were 8 degrees, again, about the sort of, the image circle was about 8 degrees, similar to the setup I had at the beginning. This data were from 39 subjects. We've since done this on many, many subjects on the web, and it's very, very consistent both ways. There were roughly 23,000 classifications made. Again, performance is D prime. That takes into account any biases and subjects preferring one object or another. It's meant to be a pure measure of sensitivity for those of you who do psychophysics. Okay, so here's kind of what we end. Just to give you a feel for this, here's one task. So I think of each of these squares as a task. So it has a D prime, it has many images associated with it, and this is a summary measure of performance. So up there is a low variation face task. And what was implicitly being done, now the humans were doing this in an 8-way version, but what's implicitly being asked of the human is, here images are faces that you should have called faces, low variation, see they're all front and center looking right at you, different backgrounds, and then there's a not face. So there's all the other 7 objects, and they had to kind of call these faces and call those not faces, and you saw they did that quite well. They would generally get these right. And so this is one, that's how we get 1 D prime, 1 point up there is thinking about a task like that. Here is high variation faces, so the faces are more twisting around here in the latent parameters, as are the objects as well. Okay, so you get another point. It's a little lower. You see the D prime is probably about 3 or 4. Here's another, this is beetle. Tell me which car is it? It's the beetle and not these other types of cars. So it's a little more challenging of a task. Again, these are small and hard for you to see. I just want to give you the intuition that they saw hundreds of these kind of images, and this is a summary measure of their performance. So the two key points here is the pattern of performance across humans is highly reliable. So this is not noise here. This is an average of a bunch of subjects, and we looked at inter-subject reliability measures, and I could pull that all out for you. But this pattern in these particular images and tasks are highly reliable. And you can see it's not that humans are perfect, right? These are not all red 5.5 D prime of 5. So those of you who think humans are the best vision system on the planet, yes, we may be good, but we're not perfect, right? So, and the difficulty, of course, depends on the information in the images and so forth. So this pattern of difficulty that's stable and not flat at 5 can be used to help us infer what kinds of codes are the humans using to produce this behavior. So we're going to use this variation in these numbers here, these color changes, to help us to say which kind of codes could be explaining this performance and what kind of codes are likely not explaining this performance. That's the way the game is played that I'm taking you through. Okay, does that behavioral data make sense to those? Yes, okay.
So can you maybe comment on why the performance, so the subordinate, I can see why that would be harder than basic, but it seems strange to me that there would be really chance performance or close to chance performance.
Yeah, because humans are supposed to be perfect at faces, right? So, but come on, face 1 and face 2, you saw those are synthetic faces without hair, right? I mean, when we talk about face recognition, there's a whole game to talk about there, right? Infinite number of tasks. Our tasks were hard, right? We don't think it would matter if we put humans in with hair, but you can see when you're asked to just do pure shape on face sets drawn, you saw those kind of face images, it's a challenging task. You're barely above chance often on these kind of, especially at high variation, which is, lots of variation latent
So
parameters.
I guess the Z3 is a sports car, and then the rest of them are small.
Yeah, so there's of course, you're inferring there's probably larger sort of shape variation at the physics level going on here, right? And we didn't really strongly attempt to control that. These were just 3D models. Again, from the purposes of our study, we don't think that matters, but, those are...
So I guess the beetle is available in the US, but I don't know if an Opel Astra is available.
Okay, so now you're asking interesting questions about what would happen across. Now, they were not given these words, sorry, by the way, I should have said that. They were given images of the 8 objects. So at the end, they sort of saw an image and like, which one was it? And they were like kind of canonical view. That's how this was actually run. So there's no word problem here. But now that we can, returk it, we can kind of do this, were you in India, are there any variation there? We haven't looked at that yet, but we could. And you're getting to sort of interesting questions that related to this kind of monkey-human distinction, which is how much of this is learned and how much of this is just limited by some hardware that most of us all have in our heads, which is really what we're after. Well, I haven't talked about monkeys yet at all, but just... This is all human psychophysics, right? This is the thing we're aiming to explain that, again, we can talk about whether this is all object recognition. It's just our version of a task that humans were asked to do that we thought, hey, you're supposed to be good at this, and you are good at it, and you're not so good at it, and you have a pattern of performance. Yeah.
So they were not familiarized with the subordinate classes?
They were implicitly familiarized because they would kind of, when they do the task for an hour, they sort of getting more and more familiarized. We gave them a little training at the beginning. You're starting to ask about size of training effects. If you do this task for, a year, how much better do you get? And we had some of our subjects do that, and they would improve from like a 1 to a 1.5. So there are training effects we can measure, but they're small relative to the scale of this. Okay, so that doesn't mean there isn't anything interesting there. I'm just trying to tell you what the data tell us so far in this time. Yeah.
I'm curious as to exactly what the task is, and you expect different performance for different tasks.
I certainly expect you to get a different D prime number out depending on your task. And there are really interesting questions there, and that's exactly the direction we'd like to go. If you change the task, at some point what we think is going on in the brain, at some point you start tapping different neurons in your head, right? I mean, this is what the ventral dorsal stream division in the brain is thought to be about. Like, am I reaching for something versus am I naming it? And those are ideas right now, but this kind of approach and then asking those kind of different questions would allow us to then say, I'm going to show you IT neurons can produce this pattern, just to cut to the chase. But maybe IT neurons won't produce a different pattern that would emerge if you ask the question a different way. Right? And that is falsifying an idea that IT doesn't seem to directly support this other task, and that's progress to me. And that's sort of where this strategy would lead you. Right? The idea is to falsify, hey, I think IT does this, but then later say, but it doesn't do that. Right? And that we can do that with these images. We haven't done it yet. But that's the nature of this kind of setup here. That's why I'm calling this 1.0, because it's just our first, it's not the end, it's the beginning. Okay. So let me sort of jump ahead, because I guess I have like 10, 15 minutes at most, right? I was 5 minutes late, but because I just want to take you through the results of this, and then I probably won't have time to talk about anything else. So since we've gone through a lot of, okay, so we had to, we recorded a bunch of neurons just for the aficionados. This was done with chronic arrays implanted deep in IT, which is, many of you might know, is kind of challenging to get them down there. I'll also say that we've done analyses on the selectivity properties that we get out of these units. And some of these are multi-units. I mean, a lot of this analysis is going to be multi-unit data, again, for the aficionados. But if you look at the selectivity properties, those are very similar to our sharp, extracellular, nicely isolated units. And that's another interesting point of discussion. But this is just to tell you how we get the data that I'm going to show you for this particular study. Okay, so we have these arrays. We recorded in V4 and throughout IT. So we have data from along the ventral stream, no V1 or V2 recorded data. The idea is the monkey's fixating. You've got arrays. This is meant to schematize active neurons. And again, the monkeys are simply fixating. You saw the images coming up 100 milliseconds on, 100 milliseconds off, randomly interleaved, so we can collect the images, the data rapidly. Think about one neuronal site shown here. Here's the kind of data you get out of a site. Those tick marks again up there, those are action potentials from one site. Here's 8 images repeated many times. You can see, again, similar to what I showed you earlier. We showed hundreds or thousands of images. Lots of details for the aficionados that I won't take you through. Again, the animal has no task other than to fixate. For simplicity, just think of counting spikes in, again, a 100 millisecond window, in this case 70 to 170 milliseconds. That's now collapsed, like I did earlier, into a scalar value for these 8 particular images. So you can get a sense of green means high activity, black means low. We had many different images in many different categories, showing you more of the data. There were actually 6,400 images, or 640 images in this case. Other data sets had 2,500 images, so we showed thousands of images roughly. You can see there's patterns of mean response at one particular site. This is just one site in IT. We had, of course, lots of sites, many of these collected simultaneously. These are 128 simultaneously collected sites. So this is just to give you a feel for the kind of data we're talking about. Thousands of images, hundreds of neuronal sites. So here we are, 168 IT neurons, and that's one image, vector, response, average response. Here's the responses to like 8 images. Again, we had something like 2,500 images shown here, so you can think of this as a big array of data. This is the kind of data that I'd like to try to release to the community. You have this sort of responses across these particular images. So this is our kind of top-level view of IT neuronal responses. And our goal now is to say, how can we read this IT and compute performance on all those tasks that the humans had done? So we did the same trick that we'd done earlier. We're going to compute a performance measure, and we're going to do this by putting linear decoders on the IT. We think again of that as a proxy for downstream brain areas, something that downstream neurons can easily do. We use a linear classifier. Many of you are familiar with that. We learn the weights from some training data, and then we compute the D prime on new, i.e. cross-validated images. And so, for instance, if we wanted to ask how well does this population support a task of face versus not face, we would train on images that look like this, faces and not faces. We would then compute a cross-validated D prime performance, and that's one number, D prime for faces, that is the predicted performance for one particular task given these images. And so that's a color value there. We do this again for cars and not cars. And of course, we can ask as many tasks as we want out of those images. We can't make new images because we've already collected the data, but we could ask latent variable tasks, for instance, about those images, and we are doing that. But this is just identity tasks task. We're asking the same, each of these squares is a task of saying, is it a car, is it not a car? It's the same kind of task that you saw earlier with different levels of variation. So this axis is identity, and this axis is level of variation ranging from low to high. And I'll show you that more clearly in a minute. We had this predicted performance pattern from IT. We had an actual performance pattern that I showed you earlier for the human. That's the same data I showed you earlier, a subset of it. And then we can ask, how well do these things relate to each other? So we're really asking, how similar is this pattern to that pattern, now laid out as a one-dimensional bar? So each of these is the performance d prime predicted on a single task, and there's something like 72 tasks in here. And so we like to compare these things directly. But before I do that for you, I want to first remind you that this is one particular way of reading one part of the brain. So we call this one putative unified code. And it's unified, meaning it's being asked to do all 72 of these tasks. But there are many ways you could read from the brain. You could read out of IT differently. You could use more neurons. You could read out of V4. You could read out of the dorsal stream. There's lots of things that can be done. So again, this is just a strategy, and I'll show you how we've expanded it so far. Here's the human target data. This is what the human does. Again, quite reliable pattern on these tasks. Here are the codes we've tested so far. So we can take IT data, build codes off of them. V4 data that we collected, build codes off of them. Computer vision features that we implement in the lab apply to the same images. V1-like things, which are simulations of the early part of the visual system, the raw pixels themselves. We can then do things like varying the time window at which we count spikes, at least for the neuronal data. So there's temporal things. We could do fancier spike codes if we liked. We haven't yet. We can do things like vary the number of units that we include, from very small to very large. We can use different types of decoders, not just linear decoders, other decoders, different ways of learning those decoders, different amounts of training examples, and so forth. So all of that are sort of parameters of the way we read the brain, including the substrate, the way of doing the time, and the way of reading it. Okay, so we think of the combination of all those parameters as one particular code, right? So it could be pixels with an SVM with 10K pixels. And that produces, in this case, a response pattern, predicted response pattern that looks like this. So just to be clear, what's being done is the pixel representation is being asked by an SVM to optimize performance for each task, and that's the resulted predicted pattern. It's not trying to fit this data. It's just trying to do each task as well as it can. And then here it is for V1-like. Here it is for V4. This is further up the ventral stream. Here's one IT code. Here's another IT code, another. So these are different ways of reading IT. And I hope you can see these patterns are starting to look more and more like the human pattern itself. And in fact, that's what I'm going to quantify for you right here. Let me just sort of, I think this is more cute, so I'm going to skip through it in the interest of time. The way you should think about this is there are many points of possible tasks, 72 tasks on here. What we're looking for is a relationship between mean human performance d prime and actual prediction of the code. We'd like them to all be on a line. We could imagine a code that has poor performance, which we'll call on all tasks. That'd be a poor code, regardless of what the human does. There's a code that would be really great on all tasks, even tasks that humans are terrible at. I call that a DARPA happy code because it would do things well, but it wouldn't be human-like. There's another code with the neuroscientists would be happy, which is highly correlated, but maybe is low performance, so maybe it doesn't make DARPA happy, but it's the correlate of object recognition. That's a neuroscientist's way of speaking. Of course, what we want is that one in the upper right trial, the true code. And the reason I'm setting this up for you is because now we can sort of talk about the two ways that we're going to compare these codes with the behavior. One is the consistency, which is the pattern of performance that you've been seeing graphically, and that's on this axis here. The other is the mean absolute, which is more like the DARPA goal. So the goal is for us to be we can have measures of mean performance, which you can think of the slope of this line, and you can have a consistency, which is like the correlation of this line. And that's exactly how we kind of measure those things. And different codes that I referred to would occupy different corners of this space here. Okay, so that's the setup. We can think of codes that can get up in the true there to being passing the Turing test for object 1.0. Object recognition 1.0. Here is this axis, the consistency axis. This is just that y-axis here. That's the pattern. So again, human-to-human consistency, it's quite good, as I referred to earlier, but it's not perfect. So you can see it's like 0.9-ish something, right? And there's some range there. So you can say any code that gets into there is sort of a viable pass of a Turing test and consistency. Here's pixel-based codes, a few of them, V1-based, some computer vision codes. I could take you through these features. Many of you know some of them. V4 reads, so that's real neural data from V4. And there's some reads out of IT. And the key thing you should see from this is that there are some IT codes that when we build them, they just say, do as well as you can on this, do as well as you can on this, do as well as you can on this. They automatically spit out a pattern of performance that looks indistinguishable from the human pattern of performance. So again, they weren't fit to match the human. They just, when you fit a decoder on them, it ends up looking just like a human in terms of its pattern. And that's not true for earlier visual codes and not true for existing computer vision codes. And that's the lever that we want to use to search through what is the right code in an artificial sense. So that's exciting to us that that's still there, which is what we'd hoped, but we never seen before. But it's also true that not all ways of reading IT produce that. And that's also powerful because it starts to allow us to take apart what is the time window that matters in IT, what kind of codes matter. And we can push that further and further with more and more data. Okay, so now this is just the pattern of performance. Can these explain that DARPA level absolute performance? And the way sort of to do that, I'm going to come back to this 2D plot. So here's consistency, the axis I was showing you earlier. This is absolute performance. And many of you know you can often increase the absolute performance by increasing the number of features. We can see that in our data here. So this is the number of neurons. So here's an increased number of features on IT. And you can see that as you get to about a few hundred, you get to the pattern of performance match. And then roughly around 500 or so IT features, you can think of 500 neurons, you're matching in both absolute level of performance and pattern of performance the human ability that I showed you earlier. Now this is not true for a nearby visual area, V4, that feeds IT, so you can see it doesn't actually do that. And in fact, if I take this, we tested many codes, as I referred to earlier. All the blue codes are reads out of IT. All the green codes are reads out of V4. Computer vision models are in red. V1-like are in black. And you can see that some ways of reading IT pass both of these tests, and most other codes are essentially falsified by this kind of analysis. So there's good news. If you believe in some of these, there's bad news from a, hey, you're not a human point of view. That's good news from a science progressing point of view. But there's also good news that there's some ways of looking at IT that we can explain object recognition performance. That is, these codes up here in the top might directly underlie object recognition behavior. That's the inference we make from these kind of data. Remember, these are primate reads, non-human primate reads explaining human performance data in both absolute level and pattern of performance. For the computer vision aficionados in the room, I want to point out that this is non-trivial from a performance point of view. The number of code elements, again, you only need roughly 500 features in IT to get here. V4 could, we think, eventually get there with many, many features in terms of absolute levels of performance, not pattern. And computer vision codes there, this is a log scale. You need many, many, many features that we think extrapolate to get anywhere near the levels of absolute performance that humans have for these kind of models. Okay, so just throw that in there. I think I'm sort of at the end of my time, so I'm just going to sort of give you a wrap up here. So there's spike count codes that are easy to read in IT. They're produced automatically and reflexively. Again, the animal is not doing a task, not being told to attend to anything. His ventral stream is just computing these features in about 100 milliseconds. These patterns, when read in some ways, as I've showed you, linear classifiers with certain time windows and so forth, can fully account for behavioral performance, at least in object recognition 1.0. And we also, as came up in discussion, we have some understanding of the relationship between what the single unit properties of those neurons are and these population reads. But I didn't talk about that at all today. So what this work, I think, contributes to is we have a view of what the solution and the nature of it might look like at the top in terms of explaining this kind of behavior. And now the hard question remains is how do you actually produce that phenomenology that we can just directly measure? So I was going to give you a 3-minute tour of years of our experimental work trying to go there, but I don't have any time to do that. So I at least got through what I wanted to focus on today. And let me sort of give you the highlights and then the wrap-up slide, and then we can take questions. So for those of you who want to get into our modeling discussion. So invariance is the crux problem, that what we think is the engineering hard problem that the brain has solved. Some IT population codes are sufficient to account for unfettered human object recognition performance. Again, these were durations of images that were comparable to natural viewing. They weren't masked after one frame or anything to kind of bring the human down, even in these engineering hard conditions. These sufficient IT codes are computed reflexively, I say, meaning automatically, passively fixating animal in 100 milliseconds by this engine of the ventral stream. The key algorithms, the processing that's happening here, lives, we think, somewhere between V1 and IT. You saw the performance of these codes and some dramatic increases going from V1 to IT. And there's details in there that have to do, I'll just say them for you. So we know that we think that performance gains are gradually built up. This is maybe not surprising. We have measurements of that. And we think that unsupervised learning, and much of our lab has been devoted to studying that, instructs the building of those transformations. So again, we're trying to get at the mechanisms to get that phenomenology. I didn't have time to tell you about that. And we're using these neuronal constraints, these measurements of IT and V4, as sort of a constraint or a regularizer on a large class of models that we're trying to screen that are putative algorithms that have this structure to say, can we discover algorithms that not only lead high performance, but that are matching or at least consistent with the kind of neural data that we've been collecting. And we're hoping to show that that strategy leads the field more directly to finding good models, even for recognition tasks, even if you originally, maybe some of us don't care as much about neurons, but I certainly do. But we want to show that those neural constraints actually add value to the search for algorithms to produce good representation, good performance. And again, our data will let us know when we have an algorithm. The neural data that I showed you will let us know when we have an algorithm that is not just kind of quote bio-inspired, but is actually kind of explaining the neural data as well. And we will continue to work to gather more and more of that data to act as a stronger and stronger constraint. And I'm interested in synergizing that with the field more broadly of computer vision. And I know I alluded to this at the beginning, but as experimentalists, one of the things we need to try to do is not just show these strong correlational measures, which is, hey, this code is consistent with explaining that behavior, but ultimately have interventional techniques where we change the activity of those neurons and show that they produce predicted changes in the animals. In this case, it would have to be monkey behavior, and that's something that the experimental lab like mine should be able to contribute to, and that's something that we're working on. So hopefully I was able to connect to you how we are trying to think about this problem and how it might inform those of you, especially working in computer vision and object recognition. And the folks who did the work are mostly shown here. I especially want to thank Najib Majaz, who's here, and Ha Hong. They did most of this coding analysis along with Ethan Solomon, again, an undergraduate in the lab. And again, thank my funding agencies and thank all of you for your time and giving me this chance to tell you about our work. Thank you. So I'm happy to take questions, and I apologize I didn't talk about our modeling or unsupervised learning or other things that are going on in the lab, but hopefully I've given you at least one story that you can take home. Yes?
It's sort of always assumed in the beginning that object recognition all involves attention and focus and all that. You seem to be suggesting it's very reflexive, passive. I wonder if it would even work in the periphery. You do the same experiment in the periphery.
Right. It's a great question. Again, we built this experiment with this. The images were 10 degrees. We didn't actually put up full scenes, but you'd like to think that, yeah, at some point it won't work, and that would show what I said earlier, that I think IT is focused based on receptive field analysis. All IT neurons generally cover the fovea, the receptive fields. So there's a strong foveal bias there that you would start to fail at these peripheral tasks. But I don't know. Well, yeah, you certainly, and then that's when people start talking about, you're going to have the dorsal stream coming into play, motion detection from MT. There's sort of things that you're going to want to use other parts of your visual system to help you. It's not just IT, right? So that's back to this broader discussion of asking other tasks on images, and then, in the broad sense, if we could record we as a field, all neurons in all parts of the brain for a large set of images, and then ask a number of tasks, we would be able to map these things more tightly. We could extend this to the periphery. It wasn't our first tech, and I mean, the attention discussion is an interesting one because that is, I alluded to that. It's something that comes up a lot, which is, wait, what happened to attention? And I don't know. I'm not trying to argue there is no attention or anything like that. I'm just saying in these kind of conditions where you ask, here's 8 objects, there are effects of attention that can be measured, but you have to put those in the context of what is making the problem challenging from an engineering perspective. And this is saying we think most of the hard work is done in this sort of more automatic mode. It's not dismissing attentional work. It's just saying, look, here's how far you can get with this, and we're kind of parsimonious, null hypothesis type people, and we'll just keep doing that until maybe it breaks in the periphery, as you say, and that might be a great next experiment to try. But that also depends if you want to politically fight the battle of attention or not, or what does that mean, and that's a messy kind of, it's a difficult area to kind of argue in. It's sort of well tread in many ways and not well operationalized. So we've sort of avoided it so far, but we'll have to get there at some point. It's difficult. Many people mean many things when they say attention. And so to make a kind of clear progress on that, I could use your help or others to tell us, like, what would be the definitive experiment? Because it's hard to get consensus on that, at least in my informal sample of people. You don't look convinced.
Well, I'm thinking in the focus area, there's both attention, but also extra hardware. I mean, clearly things like color or something.
Yeah, okay, right. And we didn't even include color, as you saw in these images. We purposely avoided color, right? So these, it's certainly if you're searching and you say that brings into feature attention, I'm looking for something red, you put in that bias ahead of time and say, how well does a human do in detecting apples when you've told them it's going to be something red, and you do tasks like that, then their performance will get better. That's what the attentional literature shows. And so, but the neurons, they're just passive, so they shouldn't change. But one of the intriguing things is we like to think about, well, look, we can retrain classifiers with, now let's it's a multi-way classifier problem, so this is sort of a late selection attention model ultimately. Let's throw away all the classifiers that are trying to do the non-red things, ignore them, so their noise and sort of voting for maybe I see something goes away, and that might explain, we could get patterns of performance that are predicted in that regard, right? But that's an argument that all the attentional effects would be sort of downstream of these neurons. But that's one way we could easily extend and try to connect attentional effects to the things we've done here. And the next would be, that's an easy thing to do. Next would be the standard thing, which is try to train the monkey, which Ed and others have done, which is a very, very hard thing to do, and then get the monkey to do attentional switching. And that would be that's the hardest thing to do, but at some point you might have to do that to explain everything. That would imply that in the human, again, that their attentional effects have filtered down to the level of, say, their V1 neurons, and their IT is in a totally different output set when you engage that. And that's, as you know, still a debated question. There's no debate about whether attentional effects psychophysically. It's a question of at what level and how strong are these impacts, right? Because when you measure those things in neurons, they're usually 10 to 20% modulations in the neurons, and they're not extreme changes in the organization of the selectivity of the neurons. So from these kind of population decodes, we don't think they're going to be strong effects, but we haven't looked at that yet.
Talk about the engineering. So all the neurons can do is local dot products and then nonlinear filtering, and then at the next layer you do that again, right? So at the 4 or 5 or 10 layers, you end up just doing that and having to figure out what the dot products are and what the nonlinear filtering is. And that's it, right?
Well, yeah, that's a standard. That's the standard feed-forward kind of hierarchy. You do something nonlinear like normalization. You pick some weighted filter sets like Gabor's or V1, some activation function of the nonlinear, maybe some more normalization. These are all nonlinearities and linear dot products, and that's the sort of standard feed-forward class of algorithms that we've mostly been exploring. And yeah, I mean, I'm sure maybe you can repeat your question, but so that's the class of models that. If you believe that, it's right. I'm with you. But again, we sort of see the different levels of sort of argument here, right? The data that we have sort of argues that, look, 100 milliseconds, something interesting is produced, and probably that's a series of steps, as already been alluded to, normalization, dot product. Let's discover that, and I'm on board with you. Let's find that. Let's use the neural data to discover it. But others are saying, well, no, that's never going to work. We have to do some top-down inference, some other feedback circuit. And so that's kind of where I think our data are valuable to contribute to that discussion, right? So I'm on your side, but not everybody is. Let's put it that way. I mean, that's, of course, again, it gets into the deep notion of what is object recognition. There are going to be some tasks where that's not going to work. And, it's just like the human will fail at Where's Waldo, right? I show Where's Waldo, was there Waldo or not? Human can't do that in 100 milliseconds. So it's that core recognition domain that sort of we're focused on that I think is of those type of calculations. And then there's this next level question of how much of vision is that, right? I mean, when we're in our daily lives. And I don't think it's hard to know the answer to that yet. But I'm agreeing with you, but also sort of trying to point out the challenges. I mean, these are the class of models that we actually work with, right? Where we stack repeated, what we do is we take an idea that tries to untangle manifolds locally, and we stack it horizontally, and then we stack it vertically. It has some parameters associated, but we repeat those parameters up and down for an iterated stack, exactly as you say. And that's a large family of algorithms that we think there might be a really powerful answer in there. And we're searching that space right now. Some may say, well, that's fruitless because you don't have any, feedback, inference, some other magic like that. But again, as engineers, we're like, well, we're going to just try, and that's our bias. Okay. I'm not trying to be dismissive of the other view. I'm just saying here's something that I know how to do. Yes.
So if I understood what you're doing is you're using the human performance to provide interpretation of the data that you're collecting from monkeys.
Yes, as a interpretation or a constraint on what kinds of neural activity might produce that human performance. Right.
So then to understand the neural response of this part of IT of the monkey, you're looking to human performance and you're looking at this match.
Right.
So it's important then to understand the human performance. So what have the humans, what have humans actually accomplished in this task? So what do they achieve in terms of the visual processing in this task? Because that's reflected in their primes, which are then matched to the monkey primes. So I'm wondering, humans, these for the faces and for the subordinate items, for the highly variable items, performance, as someone else pointed out, isn't really great. I mean, it's not terrible, but it's pretty, it's pretty iffy. And so, that if you were just looking at the human behavior, you might say, well, in this task, under these conditions, which are difficult, humans aren't really doing very good invariant perception, right? Invariance has been well achieved in these tasks. So if the humans haven't achieved sort of invariant perception in this task, but yet, their codes sort of match the monkey codes, then how confident can we be that these neuronal populations in the monkey actually achieve invariance? Because they're matching human performance, which hasn't achieved invariance. So
Right.
The conclusion that these monkey codes actually aren't doing very good invariant perception.
On some of those tasks. That's a fair interpretation. So you could say, well, look, the logic usually goes, oh, humans are magic and they do invariance. But then if you measure it, they don't in this task, right? And that's partly what's interesting. Now I think the follow-up here is we should ask other questions of the humans that try to expose where they're actually good, better at the faces than we've exposed so far, which leads to the attentional questions and the where's Waldo and the sort of, leads us outside the domain of what I'm calling core recognition, sort of this earlier discussion. And I'm happy, I think that, and then that kind of brings in the community that, in a broader sense. I mean, we just did this as a sort of first pass, 100 milliseconds, because that's why I call it core recognition, what can you do in a glimpse? But we could still add hair and things. But I mean, I think those are great next questions. And it comes down to this is where I need help from the community. I mean, we can collect neural data, but we need kind of people who want to operationalize the human tasks in a way that the computer vision community and the psychophysical community are resonant with. They say they view as actually a fair definition, and rather than everybody saying, no, I think it should be like this, and whatever, pick away and measure it, and then we can make progress. But there's more.
I was just trying to understand how we should interpret the results in terms of what they mean about the monkey neural encoding. So I wonder if you I think the method is really fascinating. I think it's going to be productive. I mean, it is productive already. And so there are a lot of other things one could ask. And I wonder whether if you did use a task where humans were achieving object invariance, whether you'd get as good or not.
Yeah, I mean, that's a great question. And that's actually our next test. Is that as we want to break, now we say, here's a code in IT. We don't want to stand up here and go, this is it, and explains everything. Let's go home. It's now let's try to break it. Right? And that's sort of where the attention, let's try the periphery, let's try the longer time to view faces. I mean, try inspiration from the ideas that you're saying, but to do that we need to then, define it and then measure it, and then go ahead and then measure the neurons. But that strategy, that's again Vernon Mountcastle's strategy. It's just extending it into this complicated domain. And I agree with what you say, and I think that's where we'd like to go. There's just so many ways to go, and that is an axis that we will try to push on. But I'm also saying, I could use help, right? There's computer vision image database questions, then there's psychophysics, how should we measure human performance questions, and if the community could rally around both of those things, we're happy to try to collect the neural data to say, can these IT codes explain that, how much of that? And that would be progress. Even if it's not all of it, that's okay, that's the result, right? But we need all kind of, the word is operationalize, and not. And I'm not pointing at you, I'm just saying that, we have to, as a field, move to measurements of such things. And people aren't usually rewarded for characterization measurements, but it's becoming easier on the Turk, right? I mean, psychophysicists are often rewarded for an idea inspired by the psychophysics rather than just give me that big set of data matrix that we need as a constraint, right? That's not a paper, right? That's not an idea. But that's the kind of data that we as a field need to do this kind of progress. So I don't know. Thanks for giving me the chance to give my little spiel there. But yeah, I don't know who's next. There's sort of one way up in the back, and then me. Right. So you're getting to the thing about, like, if there's a model, let's say it's Hmax or whatever the model, and it's sort of trying to do recognition, and it's built in a bio-inspired way, so it has a feature set that's putative IT. Now, the best comparisons of those models with neuronal data have so far been done by Nikos Chryzgorte, and using sort of distance relationships, meaning you have images in the high-dimensional space and saying, look, the model predicts these images should be close and these should be far. And he's shown that using both IT data and fMRI data, that most of those models aren't anywhere close to what the neuronal data say. That doesn't mean they're not useful ideas in those models. It just means none of the models have sort of met the bar of the data we already have, let alone the data we're going to collect tomorrow. So, but that line of thinking is exactly what we're trying to do with these models. Is not, they can't just perform, but they have to. That's what I meant by being regularized by the data. If they're not actually matching, then we know they're wrong if we really want a true bio-inspired model. And metrics for comparing models with neurons are sort of a big part of what we're trying to do. But so far, to answer your question, Hmax, none of them succeed in explaining the data in that strong way. They can overfit a single, give you an IT neuron and say, fit it. But that's not the right way to phrase the problem. It has to sort of emerge as part of the principles of the model. It should look like IT, right? It shouldn't just be overfit. Because the data are limited ultimately, so it can always be sort of overfit in a way.
Sorry, yeah. Just to follow up on that, the experiments that you mentioned, if you take IT neurons from your brain and from my brain, do they agree on the similarity there?
Yes. Here's actually, I think I have that slide here. This is Chryzgorte's review of that. And this is sort of lack of fit here, right? So here they actually have Hmax. So this is, they showed, for instance, this is monkey and human. This is distances among images. That's what's the color in those matrices. The human is actually fMRI data. The monkey is IT response data. And at least at the level of, what you see there is a big dominance of faces, as you might imagine from the human fMRI data. But there's some similarity that you can get between the monkeys and humans, and they also show human-to-human consistency. Again, that's the level of MR, so it's a blurry version. But you can also see levels of consistency IT to IT. So we take, for instance, a bunch of IT neurons measured on day one and another 100 neurons of IT neurons, and we can do measures like this and say, how similar are they? Which is a question of, have you sampled IT enough, right? If it's a million-dimensional space, you might predict that 100 neurons here, 100, maybe they look nothing like each other. But the fact is, when you get to a few hundred, they actually look quite similar in terms of layout of images. Now, quite similar is a correlation that's greater than 0 and less than 1. But it's like, it's like 0.7-ish in our hands, right? So, what does that mean? It's not, 50% explained variance. So I'm telling you, we've done those kind of comparisons. They're not perfect, but they suggest that a few hundred sites already gives you something that's reproducible on neurons. And you should read this paper if you want to see about humans, because we don't do human MR. But this is basically also a paper showing here's many different models trying to show fit. And this would be perfect. This would be as within the noise levels of the data, I think, of this line here. And they're showing that all these models tested so far aren't anywhere near what the data self-consistency should be, which in this plot is like down here. This is like a lack of fit plot.
That's not just because the model is exposed to a different distribution.
Nobody knows. I mean, all they're doing is running that model on those images, right? So, I mean, again, we're into debates about, it's not like the ideas of Hmax are wrong, right? I mean, implicitly, they sort of capture a large space of ideas. It's just that the particular implementation is not correct, right? And how we, as a field, use the word model is, I think, a thing that we need to do a better job at. To me, a model should be, here's an instance, a prediction. It will be downloaded, it will produce a response pattern for this image. It's not a set of ideas. Those are ideas. It's, it's a particular instance. And then model 1269 has been falsified. 1263 still is in play, right? That kind of spirit, right? And that's what we're trying to do with the neural codes. You saw our gazillion codes. I'd like to see gazillions of models, and models in an implemented sense. And we can screen through them in that way. And how we use the word model is a big part of that. Maybe we should call it something else, model instance. So some model instances of Hmax have been technically falsified, but Hmax as an idea is not falsified. In fact, I believe in it in the same sense that we discussed earlier with sort of nonlinear linear stacks, right? And that's the class we're exploring in a way. Yes, I don't know. You guys have to cut me off, right? Because I'm well over your time, but I'm happy to stay as long as you want. So it's up to you guys to just walk out, or yeah, go ahead. Right.
Maybe we're looking in the wrong direction. We should focus on what we have to do and logical process involved. So instead of worrying about device, we can take forward over skills and go over limitations and forget about what the brain does. So I want to know what your answer is. Personally, I don't agree, but I want to know what you calculate.
My answer to that right now is we're at a nice time in history where even if what you said is true, the neuroscientists and the NIH need to know how this particular piece of hardware works, right? So there's still a drive to understand it. Even if we had imagine we had a bunch of models that are all successfully better than humans, walked around, beat us in all these tasks, and us poor human hardware is well left behind, like doing calculus, playing chess, the things we've already been beaten at. There'd still be a neuroscientist's goal, and this is how a lot of neuroscience works, is like I need to map the instance, which model fits which component of this particular piece of wetware because I need to fix it, cure something, intervene, augment, right? So that will always remain from a neuroscience perspective. We're in an interesting time in history that those models that I referred to don't yet appear to exist. We still win. So there's a chance that what the ideas about wings, to use your analogy, that we can get from the brain might inspire maybe not the exact model, but still might drive the creation of models that will be of interest to someone like Google or DARPA that maybe doesn't actually care about fixing a brain, but wants to succeed in having a drone recognize objects on the ground. So I think this time in history means both options are open to folks like you and me, but maybe in 20 years, only that neuroscience pure connection option will remain. And I think that's kind of my prediction, is that soon it will look more like the neuroscientists have to clean up the mapping problem, and there'll be less time for inspiration, because engineering and Google and so forth will march on independent of this neural data. And I'd like to try to inspire it and make it go faster, but I can't predict whether we'll actually be able to do that. As you said, a bunch of engineers working hard enough could overrun neuroscientists doing their best, because there aren't that many of us thinking this way. They're mostly thinking about connecting to the actual disease process or something more along those kind of standard wetware lines rather than computation lines. And I don't know. I'm trying. That's our background. We think we can try, but I don't know. Does that answer your question? Well, right. So that's where I can use help, especially computer vision. Tell us where you're stuck, right? And you're not. Everybody said this to me early on. They're not stuck on translation. They can do pan and scan, right? So we don't need to worry about translation. We should worry about, clutter or pose. Those are the ones that we're stuck on, and so we should study that directly. And I can, that would be one way to go. But I guess we kind of view, the brain as sort of the more your algorithm diverts from the brain, then the less I kind of follow that, because I feel like, well, the brain has solved translation probably in a completely different way. So it's like you've already kind of left the boat once you sort of do that, right? You're using your engineering tricks and you're building models that are trying to be DARPA-like, but you've given up on you're not actually playing the bio-driven game anymore, right? So, I like to think that the way the brain solved translation might inform how it solves pose, right? It's not, so the brain has to solve translation too, and yeah, it could have found this other trick, but maybe it found a deeper trick that does all of that. Right? That's sort of the hope, right? That it didn't fall, there wasn't a heuristic that we as an engineering community stumbled upon, but there's another deeper heuristic that solves all of it together, right? And that's why we think it's still valid to study things like translation and scale invariance in the brain, because it might connect to that idea. But we're all just guessing, right? We're scientists with intuitions, and there's I can't tell you that's the right answer. I'm just giving you my view on it. Yeah. Right, so you would predict a match at, say, the output stage. Let's say IT might make you happy, but you wouldn't care about the level of the details of what happened between V1 and IT.
There's still some, I mean, if you do the processing in a very different order, then maybe not, but I'm guessing that the kind of intrinsic, there might be some sort of intrinsic order of these things that needs to happen.
Mm-hmm.
And so you would expect that there would be some match between these different manifold representations as a program.
Yeah, you're sort of saying that the problem itself might constrain the model classes so much as to force them to. But that's always dangerous because it's sort of like, arguments about would we have been a different species under, the same constraints with different initial conditions, right, where somebody happened to develop some type of hardware and they build it one way, right? It seems to me not clear at all that we end up in the same path. So I mean, but we don't know, right? It's a hard question, yeah. How do we know it's a good representation for? Well, I mean, you say that, but how do we know that? That's what these studies get at, right? They say, look, we're going to read this way and ask what can happen. And you read that way, and here's what can happen. And, I think the right answer to the response is, well, no, humans, I think, is to this question up here, which is like, well, actually, I think humans can do more. Let's make the object recognition task harder. And because right now, these tests aren't exposing a limitation of that way of reading from the neuron. So our hypothesis effectively is the brain, when you do recognition, you have some downstream classifiers that have learned with some data. They're counting spikes over a sloppy window of 100 milliseconds. And that, according to all this data, seems like a viable model. It doesn't tell you how to build it because it doesn't know how to build IT. But it says downstream of IT, here's all you need to do. And you might say, no, I like to think the brain is trickier and it does some complicated thing in IT. And I go, that's great, but your parsimony would argue for me. Like that's fine, but you're making more complicated statements. You need to bring data to support that. I'm not disagreeing with you, but we can't just say we know that. We can't just, I need to know what experiment's going to expose that. Because right now, things often when you look closely, they're simpler than you might have thought, right? And I think that's a nice thing. So anyway, we should probably wrap up tonight. I think so. Thanks so much.